We can start off. Hello, my name is Carolina Sandoval,
And I am Norma Hernandez.
and today we will be talking to you about our special video project, which is the X chromosome inactivation. But before we do, let's take it back to the basics, back to genetics 101. And let's look at a regular female karyotype that's on your left side. As you can see, there are 23 pairs of chromosomes altogether, but only 22 of them are autosomal chromosomes, and each chromatid comes from each parent. So one from mom, one from dad, one from mom, one from dad, and etc. But today we are going to be focusing on this special pair down here in the bottom right, which is the sex chromosome. So for double X, it is a female, and then for X and Y, it is a male. And these chromosomes are also known as sexually dimorphic, and that's because for X and Y, X and Y, it's a male, and then X and X is a female. So it's different between males and females. And to determine sex in mammals, it is by a pair of unequal sex chromosomes, which produces genetic inequality, and it has to be compensated for to be proceeded normally. And it's leading into today's topic, X chromosome inactivation. Ooh, that's today's topic, the special topic. So to introduce you to X chromosome inactivation, it has been a problem that has been worked on for over 22 years. X chromosome inactivation is a transcriptionally silent movement. It happens when an X chromosome is permanently inactivated during early female embryonic development, and we silently kill one of the X chromosomes in the female sex. So like silence, I will kill you. I hope you get that reference, but I will let our friend Ahmad explain further. Go ahead. Thank you. So I hope you get that reference because, like, silently killing. Get it? Get it? But anyways, sorry. I did not. Okay, anyways. So you may be wondering, well, why is the X chromosome and X chromosome inactivation important? Well, the X chromosome, some fun facts about the X chromosome is that it carries about 1,000 genes on the X or in it, and it makes up 5% of the genome. And it is responsible for sexual dimorphism along with Y, as well as it comes with enriched genes that are responsible for reproduction, brain development, behavior, and cognition. And it carries at least 200 disease genes in it. And so many of these genes are responsible for neurodevelopmental disorders, such as autism and other intellectual disabilities. And it also causes the Rett syndrome that is mostly in females, the Fragile X syndrome that could be in male and female, and then the traits where it's seen as well is in red and green color blindness, which does affect 10% of all men on this earth, which is like crazy. So you can also tell the X chromosome is like 3 times bigger than the Y, and that's because of all of the 1,000 genes it carries. and the Y chromosome only carries about like 100 to like maybe 300 genes. That's like 3 times bigger. So is the X chromosome inactivation an epigenetic situation? Well, yes, it is. And that's because, as we learned in genetics, it's an epigenetic, or it is an epigenetic situation because it changes slash alters gene expression. And in this case, it has males and females express only one functioning copy of the X chromosome. And in the X chromosome, it is the only one that's capable of going through global inactivation. And you may be wondering, well, like, why is this important? Well, the X chromosome inactivation models RNA, such as non-coding RNA and epigenetic silencing, as well as there are numerous disease genes that are linked on the X chromosome. And I'm just going to take you back to a little bit of time travel. So back in history, the first time it was visually seen was in 1949 when 2 Canadian scientists known as Murray L. Barr and Edward G. Bertman saw the X chromosomes inactivation that they called Barr bodies in cats, like out of everything, cats. So in these figures right here, as you can see where the white arrow is at, there is a small dot right there next to the nuclei in figure 1, and then over here in figure 3, there is another Barr body, and Barr bodies are X chromosome inactivations, and they can only be found in female cats. If you look here at figure 2, there is no small dot next to the nuclei, and that's because this is a male cat. So although they didn't know exactly what they were looking at, they knew that genetically they could know the sex of a cat. And it's crazy because they didn't even physically ever see these cats. They just took their blood and ran these tests and learned it. And then in 1959, an American scientist that goes by Susumu Ono discovered that in rats, the number of Barr bodies parallels the number of X chromosomes. So like right here, where it's a male, of course, males don't have any. And then in females, you can see 1 Barr body right there, and then 2 right there. And then in 1961, British scientist who put it all together was Mary F. Lyon, and she was the first one to call the Barr body an inactivated X chromosome. And then she was the one who put who said that XCI was a dosage compensation, and it is the mechanism that allows females and males to equalize and have like chromosome dosage like at the same time. So the big takeaway here is X inactivation is random, and it's made by every single cell in the female body. So, well, that lets you know that every female mammal is a mosaic, because we have, or like, well, I guess because we are mammals, we have the choice to inactivate either our dad's or our mom's chromosome. So, get it? Mosaic, because females are mosaic. And you may be wondering, how does the X chromosome inactivation work? Well, it's driven by a single master locus, which is the control center, we like to call it the master switch, and it actually does not depend on any X-specific elements. And it is populated by long non-coding RNA that's made up to tiny 100 to 200 kb regions. And as you can see in this figure down here, that's where you can see them. And then the steps of random XCI There are four, but the first one is counting mechanisms, and that's when you determine the number of X chromosomes in the cell and whether X inactivation should take place. The second step is allelic choice, and it allows the embryo to pick in between the mother's chromosome. Oh God, that's supposed to say or, ignore that, or the father's chromosome for inactivation. The third part is nucleation and spread, which is when we silence the nuclei and spread. And the fourth part is the maintenance phase, but I will let Norma tell you more about that.
All right. Hi, Dr. Ray, first of all. Okay, so like Gato had briefly mentioned, the first step is the counting mechanism, and it is located in the blastocyte shortly after paternal X chromosome is activated. And the blastocyte is a structure formed early in the embryonic development of mammals. Now, rather than physically counting, the cells are actually sensing the X chromosome to autosomal ratio. So every cell determines how many X chromosomes will be inactivated by following the N-1 rule per diploid content. So as we all know, males only have one X chromosomes, so they aren't lucky and they don't get to inactivate an X chromosome. But there are exceptions to this, as we've learned in class with the Klinefelter syndrome that gives males an extra X chromosome. And because of that, they are able to undergo X inactivation. As for females, they have two X chromosomes. So, for example, because they have two X chromosomes, by following the rule, only one of them would be active. And like we learned in class as well, females with the triple X, because they have three, and if we follow the rule, they would have two inactive bar bodies, as shown in the picture right here. So what counting really is the titration of the X and A factors. So that ratio determines whether X inactivation will occur. So the X chromosomes produce numerator factors, and they are shown in the green in the picture right here. The autosomal chromosomes produce denominator factors, and they are the orange. So once these two chromosomes produce their own factors, they will titrate each other to form a blocking factor that sits on the X chromosome to suppress inactivation. Both males and females produce a blocking factor, but because females have an extra chromosome, that extra chromosome produces a competence factor. And what that basically is an extra numerator that was left untitrated and sits on the remaining X chromosome to initiate inactivation. So the location of the inactivation site is called X-cyst, and it is located right here. This whole picture right here is the X inactivation center. And the X-cyst, like Gato had mentioned previously, is a long non-coding RNA. And what it does, it codes and silences X chromosomes. So what are the molecular factors that make up the X and A ratio? So there are a couple rules for determining the numerator. And the first one is to be produced by the X inactivation center. The second one is that it has to escape X inactivation to serve as dosage sensitive readout of X. The third is it has to be diffusible. And the fourth is it has to act on the X inactivation center to induce X cyst. So by process of elimination, they were able to find that JPX is the candidate numerator. And that is produced in the X-linked center. As for the denominator, they found that it was the CTCF autosomal protein, and it is a repressor of X-cyst. And when bound to X-cyst can't activate.
X-cyst.
So how does CTCF and JPX interact? Well, first we have to know that CTCF is an RNA binding protein. So it prefers RNA instead of DNA. And because of that, their relationship is considered a functional antagonism, which is they produce opposite effects of each other. So the first step is JPX RNA is overexpressed. And because of this, it's floating around and it binds to the CTCF that is in front of the X cyst that is blocking it's blocking it from, or it's suppressing it basically. So once it binds to CTCF, it pulls it away to no longer block X cyst. And then the third step is shown right here. And basically now that CTCF is away, X cyst promoter can be expressed, causing the X inactivation. So the second step is allele choice, and it must be random, instantaneous, mutually exclusive, and irreversible. So making the right choice, how do they do that? Well, first we had to infer that some communication must have occurred between the X chromosomes, because once one is chosen as the inactive, the other one is instantaneously active. So this is, well, this they considered as a mutually exclusive choice that requires two genetic loci at the X inactivation center. So the Psi-X is the antisense repressor, and it is right here in yellow. And then the X site is the enhancer, which is right here. So they're very close to each other, and they form the allelic choice. So what we found out is actually that Psi-X is very essential for the allele choice. Prior to X inactivation, Psi-X RNA is expressed in high levels, preventing X cyst from activating. Once inactivation starts, X cyst disappears. Woo! And forms the X inactive chromosome. As for the other X chromosome, because X site, which is the enhancer, is still present, it suppresses X cyst and stays as the active X chromosome. And right here, it's already like when that has occurred. So Psi-X would have been here, but suddenly it disappeared, allowing X cyst to activate, making this chromosome the X inactivation. And because X site is still here and doing its work, this X chromosome stays active.
Yeah, and well, to conclude our X chromosome inactivation, the first takeaway that you must remember is every female mammal is a mosaic. So yes, we are indeed a piece of art. And we have or that, sorry, I lost my mind, that has inactivated the paternal chromosome or the maternal chromosome. The second takeaway is although the majority of X chromosome inactivation happens in females, yeah, it can happen in men, but that's only if they have the Klinefelter syndrome, and that's due to the extra X. And our third takeaway, the third main idea, is the X chromosome, or like the third takeaway. Sorry, I'm nervous. Is that the X chromosome does hold several gene diseases, and that could be up to 200 or more. So that's why it's important to keep studying and learning more about this X chromosome inactivation. So thank you. We hope you liked our presentation.
Thank you, Dr. A.
And always remember that there are 2 X chromosomes, but only one of them can inactivate. Thank you.